Hi everyone! Welcome to my first LV. So this video is actually going to split into two different videos because I don't have the upgraded version of this video maker. So the first video is going to be on gene level mutations, and then the second video is going to be on chromosomal level mutations. So before we start, most people think that mutations are always bad. Mutations are a bad thing. Well, that's not always true. Actually, there are a lot of mutations that can lead to good things because when mutations occur, sometimes those mutations are better for the environment or better for survival, so that can actually lead to evolution and survival of the fittest. So don't always think that mutations are bad. They can also be good. So what causes mutations? So during replication, spontaneous errors can occur from the DNA. Most of those errors are actually caught and repaired by the enzyme DNA polymerase, but sometimes DNA polymerase misses those errors and does not fix them, so then that is called a mutation in the DNA sequence. There are also environmental factors that can lead to mutations, such as radiation or different chemicals. So radiation is the process by which electromagnetic energy travels through space or a medium such as air. An example of this would be ultraviolet or UV. Another example would be gamma rays or X-rays, which can lead to cancer, and those cells are going to replicate. Those mutated cells will replicate. I'm blocking some words. Sorry. And yes, as I said, cancer may occur. So there's also some chemicals that can cause mutations, and they can cause nucleotide substitutions or a change in the number of nucleotides in the DNA. So that's such as nitrates are turned into nitrous acid, or certain chemicals used in food preservation can cause different mutations in the DNA. So other chemicals structurally change the bases in DNA, so their structures just change, causing the base pair change after replication. Here's an example. 5' bromouracil structure is shaped just like thymine, so it can insert itself into that thymine spot, and it results in a base pair change, changing the base pairing of the DNA. So here we go into our gene mutations. So gene mutations are only on a single nucleotide. So only a single nucleotide is going to be changed in a gene mutation. So two types of gene mutations are point mutations and frame shift mutations. So point mutations are mutations that involve replacing one or more nucleotides in a DNA molecule with other nucleotides. They occur at a certain point in the DNA sequence. So you can kind of remember a certain point mutation. And then it only affects a single codon sequence. So those 3 letters in the codon sequence would only be affected. An example of this would be a substitution mutation. So a certain one nucleotide is substituted in for another nucleotide. The next one we're going to go over is a frame shift mutation. And these are mutational events in which a number of bases other than multiples of 3 are added or removed from the DNA, causing a shift in the codon reading frame. So the whole sequence is going to be shifted one way or the other. An example of this would be deletion or an insertion. So if you delete a nucleotide from the sequence, it's going to move everything over, and also if you insert a nucleotide into the sequence, it's going to shift everything over to the other way. So back to substitute point mutations. They are called missense mutations, which alter a single amino acid. So one or more nitrogenous bases are removed and replaced by other bases. So the correct base is removed and replaced with an incorrect base. The base number does not change. You still have the same number of bases, but the one single base is changed. A silent mutation occurs if the change in the DNA nucleotide does not change the amino acid sequence. So there are amino acids that can be coded from multiple codons. Multiple codons can code for the same amino acid. So even if the one nucleotide is changed, it still would code for the same amino acid, so that's kind of like a silent mutation. It still makes the same amino acid even though if there was a change in the sequence. A nonsense mutation occurs if a change in the DNA nucleotide inserts a stop codon and prematurely ends the sequence. So the protein would be stopped early because the stop codon would be inserted, which would lead to more problems such as function of the cell. And yeah, so if you look up here at this sentence, the fat cat ate his hat. That's the normal sentence. Then if you look down here, the fat rat ate his hat. So this C was changed to an R. So this R was substituted in for this C. So this would be called a single point mutation. It doesn't change the whole meaning of a sentence, but it changes that one codon, that three nucleotide sequence right here is going to be changed because of that one R is changed. Now down here, the fat dog ate his hat. This would be a multiple point mutation. Multiple nucleotides are changed in this situation. So it now says the fat dog ate his hat instead of the fat cat ate his hat or the fat rat ate his hat. Here's an example with an actual DNA and mRNA sequence. So here's the normal sequence down here. And then if you look up here, this T is substituted with a C, which then changes the mRNA sequence. Instead of it being TA, it is CG, and then which affects the amino acid that is added. It is glycine is added instead of glutamic acid, so the protein is then therefore changed. Frame shift mutations are a lot worse than substitute mutations or point substitute mutations because they affect the sequence a lot more. So an example of this would be deletion and insertion. So the cause of this causes the reading frame to shift either one way or the other. So when there's a mutation, it affects all of the codons or amino acids after that mutation occurs. So if you look up here, back to the cat sentence again, the fat cat ate his hat. So if you, that is normal. So if you remove that T, this would be a deletion. Oh, just kidding. That's done. So you are actually inserting an A here. Right in the middle of this A and T, you're inserting an A here. I can draw this. Oh, uh-oh. Just kidding. We're not going to draw it. So you're going to insert an A here, and that is going to cause the whole sequence to shift over. So instead of it being the fat cat ate his hat, it is now going to be the fa, ta, ka, tat, iha, she, ti. And then this T out here is going to be left alone. In a deletion, so you're going to delete that A, it would be the ka, ata, te, ish, at. And you'd only have 2 here instead of T, of 1 T. Move me over here. So now here is an actual sequence of a DNA frame shift mutation. So you are going to be removing this G here. So when you remove this G, everything is going to shift over to the left, and now it's going to actually code for a stop codon early, which would be that nonsense mutation, and then after that glycine would be added twice. It's going to stop early, which is not good. So if you remove if you delete that G, that's what's going to affect the whole sequence. So mutations can be repaired by that DNA polymerase we talked about before. If there are errors in the replication, the enzyme can go in there and correct the mistakes, but that does not always happen, which leads to the mutation. Alright, that's it for this video. I'm going to stop this one and then start right here for chromosomal mutations. So I'm going to end it, and then we'll go to that one.